Hello everyone, it's Mimi here. BMS working in the NHS. And today's video will be about molecular analysis of sample homogenates, which is part of the IBMS specialist portfolio in cellular pathology. So, just a quick introduction to this video. It will be short and sweet, I promise. I will discuss indicative repertoire, knowledge requirements, competence requirements, reflective statement, evidence of achievement, Q&A suggestions, supporting evidence ideas and some helpful tips that can make your portfolio really good. With all that said, I hope you will enjoy the content of my video and I hope you will learn a lot. So let's dive into the subsection. The portfolio offers indicative repertoire for this section that includes BRAF V600E, CRAS, EGFR, ROS1, T-cell gene rearrangement studies. Knowledge requirement is what it is expected of you to know and understand to demonstrate your knowledge is at the specialist level. Think about what knowledge you had as a Band 5 BMS. How would that differ to the knowledge as a Band 6 BMS? That specialist level of knowledge would allow you to troubleshoot, problem solve, verify, validate, help others and maintain high-quality service. Let's have a look at the first knowledge requirement, which is for you to know the principles of molecular biology methodology. What molecular techniques can be used to assess histological tissue? How do those methods work? Consider molecular methods like PCR, sequencing, microarrays, blotting, mass spectrometry, tissue preparation, detection of genetic mutations, nucleic acids and proteins, advanced imaging. Also consider principles of methods stated in indicative repertoire and methods used in your laboratory. The second knowledge requirement is for you to know the clinical value and application of molecular biology techniques in diagnostic cellular pathology. What is the clinical value of molecular techniques used in histology? When are they applied? Consider diagnostic accuracy, prognosis, disease identification, monitoring, guiding therapy, role in personalized medicine, cancer and infectious diseases, monitoring treatment, detecting hereditary diseases, identify predisposition and guide preventative strategies. Also consider each molecular technique used and their value, advantages and disadvantages. The third knowledge requirement is for you to know the role of molecular methods using sample homogenates in the diagnosis, prognosis, therapeutic implications of key named pathologies. What role do molecular methods using sample homogenate play in the diagnosis, prognosis, and treatment of key pathologies? Consider the role of classification and subtyping diseases, detection of genetic alterations, differential diagnosis of ambiguous tumors, detecting unknown primaries. Consider the role in prognosis like estimating progression and aggressiveness of disease, detecting micrometastasis. Consider the role in treatment and guiding personalized or targeted therapy, minimizing toxicity from drugs. Competence requirement is basically a combination of knowledge, skills, and experience one needs to perform a job to a satisfactory standard and level. Your chosen evidence should demonstrate these competence requirements. The following competence requirements need to be met for this section. Let's explore the first competence requirement, which is for you to prepare sample homogenates from cell samples for DNA and RNA analysis. How would you prepare cell samples for DNA and RNA analysis? Consider sample size, handling, temperature control to preserve genetic material, mechanical, cryogenic or homogenization of sample, lysis, extraction of genetic material, separation. Also consider different methods for fresh and FFPE tissues, use of sterile plasticware, cleaning with bleach, ethanol, minimizing cross-contamination, sample stabilization for delayed processing. What steps would you take and why? What problems could you encounter and how would you deal with them? Consider deparaffinization, rehydration, DNA extraction, RNA isolation, purification, amplification. The second competence requirement is for you to select and use control material, maintaining appropriate audit trails. How would you select control material for molecular analysis of sample homogenate? How would you maintain audit trail? Consider tissue type and availability, verification and validation of control material, selecting correct control material, commercial kits available and their control material, record keeping, traceability, audit trail. The third competence requirement is for you to assess preparations for nucleic acid integrity and purity. How would you assess nucleic acid integrity and purity? What methods would you use and why? Consider spectrophotometry for assessing purity and detecting contamination with proteins, salt and solvents, use of fluorometry for purity via comparative quantification, use of electrophoresis to assess integrity of nucleic acid either via RNA integrity number in fresh tissue or via DV200 metric for RNA fragments longer than 200 nucleotides. Use of quantitative PCR to assess functional integrity. The fourth competence requirement is for you to undertake qPCR analysis for identification of specific gene sequences. How would you undertake qPCR analysis for identification of specific gene sequences? Consider your training and competence, SOPs, methodology used, sample preparation, denaturation, annealing, extension, commercial kits available. The fifth competence requirement is for you to resolve problems associated with the demonstration methods, including root cause analysis, as appropriate. What types of staining problems can you encounter during in situ hybridization? How would you resolve them? What actions would you take? How would you communicate these problems? Consider investigation, problem identification, corrective actions to fix the issue, preventive actions to minimize problems from reoccurring. Consider possible problems with sample degradation like thermal, enzymatic or oxidative, incomplete lysis, low yield, contamination, interference, technical and methodological limitations, sample-specific challenges like fatty or tough tissues, blood contamination, fragmentation of genetic material. The sixth competence requirement is for you to dispose of waste reagents and residual material safely. How do you dispose safely reagents and residual material? What disposal risks are associated with those reagents and samples? Consider licensed contractors used by your lab, waste segregation, safety data sheet of reagents that often state disposal ways, risk assessments, local policies and regulations that are followed, health and safety risks associated with reagents like flammability, corrosivity, environmental toxicity. Also consider risks associated with disposal or residual material like human tissue. Each section also contains reflective statement which is asking you, the candidate, to summarize your role in the laboratory in the context with each section. When you complete all the subsections within this section, you will be required to write a reflective statement. You also needs to write a personal reflection on your training and why you chose your evidence. To prepare for that, write a smaller reflective statement for each subsection. What is your role in context of this subsection within the laboratory? What does your evidence demonstrate? Also write a smaller justification as to why you chose your evidence for this subsection. Also write what was your training like when it comes to this subsection. Writing a smaller reflective statement at the end of each subsection will then make it much easier at the end of the section. Each subsection also has evidence of achievement. This is a part where your training officer signs you off as competent and fit to practice as a BMS at the specialist level. Training officer will confirm that you can work in accordance with local SOPs relevant to this section. They will also confirm that you have answered questions set by them to test knowledge and skills relevant to this section. And they will confirm that your chosen evidence demonstrates your competence relevant to this section. You will need to be signed off as competent by internal assessor for this section. This can be your training officer, senior BMS overseeing this section, or a training lead. Questions should be set by the training officer. They need to be based around the knowledge requirements, relevant for each subsection. They should allow the candidate to demonstrate their understanding in each subsection. The questions should be specific and relevant to the candidate's in-house practice. They should also allow the candidate to demonstrate their specialist knowledge. If you would like to see specific example questions, please have a look at the video for trainers. Link will be included in the video description. These are some ideas for supportive evidence you will have to submit to demonstrate your competence in this area. Some more ideas for supportive evidence to demonstrate your competence in this area. Some more ideas for supportive evidence to demonstrate your competence in this area. I would also like to share with you some helpful tips that can make your portfolio look really good. Try to present the information in various formats such as tables, pictures, photographs, diagrams, flowcharts, graphs, or even videos, if you can. This nicely breaks down a lot of text that is usually present in specialist portfolios. Try to make it a little bit more interesting to the reader, which will be your training officer and eventually the assessor. Try to put yourself in their shoes. How would you like to read hundreds of pages of plain text? There is a saying I really like, and it goes like this: a picture is worth 1,000 words. I will make a video. I will share with you my evidence I used for my portfolio back in the day. Hopefully that will help. Also try to use various types of evidence, not just one type, like case studies for each subsection. You could use experiments, case studies, annotated results, annotated photographs of you performing tasks, evidence of troubleshooting, annotated EQA results, nonconformances, validation and verification as comparisons of different approaches and their pros and cons, control material production, certificates of course attendance with reflective statements, annotated images or charts, and so on. Try to bring in your personality. Think about the ways you like to learn best. If you like experimenting, perform experiments. Like troubleshooting, perform troubleshooting. Choose something that demonstrates your understanding, knowledge, skills, and how you apply this in your practice. And one more thing, keep the competence requirements in mind when you selecting your evidence. Good evidence will tick off several requirements. And this brings me to the end of today's video. Hopefully you now have a better understanding about the knowledge and competence requirement for this section. And I hope you also have a better idea what evidence you can choose to demonstrate your competence. And my final take-home message? Keep those requirements in mind when answering questions and selecting your evidence. Bye for now.